The following is an update on the research and study of LMBBS, presented by Professor Philip Beales.
Right, well, on with the programme. I'm just going to quickly run through what the order of play is for the day, so that those of you who are unable to see that will be reminded of what's coming up. So I will spend a couple of hours giving you an update on the clinical and the research components that we've been undertaking in the last over the last year since the last meeting. At 10:40, we're going to hear the first of our personal perspectives, entitled Dan the Man, from Dan Evans. 10:55, we will have the refreshments just out here in Collingtree 2 and across the corridor here. Phil's already told you about the coffee arrangements. 11:15, our guest speaker, Jim Bainbridge. I don't know whether he's arrived yet. Is Jim? I thought I saw Jim earlier. Oh, hi, Jim. Okay. He'll be talking to us. And then 11:45, Abigail from Great Ormond Street will talk about communication difficulties. 12:15, Ray's going to talk about benefits. And at 12:30, the second of our personal perspectives, running up that hill from Tonya. And then we'll have the closing remarks at 12:45. And then we'll all be ready for lunch at 1 p.m. And we promise we'll keep the time this year. Okay. So without further ado, I will kick off with an update on what we've been doing over the last year or so. So I'm going to begin with a roundup of the clinic. So just very quickly, just for my benefit, how many of you in the audience, if you could put your hands up, it's the first time at this conference? Ooh, quite a few, quite a few. Okay, well, welcome. So for your benefit, because most everyone else knows about this, but back in 2010, actually it's prior to 2010, we convinced the Department of Health in England to fund clinics specifically for patients with Bardet-Biedl syndrome. And the argument there was that you guys have so many unique problems that we needed a unique or bespoke clinic to be able to deal with each of those problems. And so what we developed was what we call a multidisciplinary clinic. So we have at least 7 specialists at each of those clinics. But we also have these clinics mirrored throughout the country, so that we have, in London, we have 2 clinics, one at Great Ormond Street Hospital, where we only see children up until about 17 years of age. And when they reach that ripe old age, they get transitioned over to the adult clinic, which is based at Guy's. And in Birmingham, we have a similar arrangement whereby we have the adult clinic at Birmingham, at UHP, or Queen Elizabeth Hospital in Birmingham, and the children are seen at Birmingham Children's Hospital. And by setting it up in this particular way, we were hoping to ensure that we get as much geographical outreach as possible. So between us, we essentially divide up roughly the 500 or so, about 470 patients who now come to this clinic. So we've been very fortunate in having the continued support of the Department of Health, which is now NHS England, who fund this particular clinic, and we're pleased to say that they're going to continue to run it and fund it for the foreseeable future. So this is a hugely positive note that I can open, I think, this particular talk on. So I'm really pleased with the way the clinics have been going, and the feedback that we've been getting, I think, also indicate that. So we've got uniquely the support of the liaison officers or the family representatives from this particular organisation, now called BBS UK, Bardet-Biedl Syndrome UK. And for those who don't know, I'm sure most of you do know, but sterling efforts, obviously, from Tonya and Julie, who coordinate those clinics, along with Chris. We've now been joined by Angela and Jackie through funding from the Genes for Genes, so congratulations there as well. And we have multiple volunteers who contribute unpaid to these clinics, including Kevin, Laura, Jenny, and anyone else who I might have met, so might have left out, so apologies for that if I have done. But without that support and everyone heaving to, these clinics would not be the success they are. These clinics are also held up. I said this last year, for example, they're held up as a real example of how you should be running these clinics for rare conditions. And many, many people keep coming to us from around the world, Canada, United States, Australia, as well as Europe, to ask us how can we share our experiences with them. We've had visits from the Norwegian clinicians in the last few months to several of the clinics in the country, and they've taken away, well, left us with a lot of positive comments, but taken away a lot of tips, if you like, to set up similar clinics in their own countries. So well done to everyone. So what we have to do as part of our efforts within the clinical service, we have to run what's called an audit day, which is a good thing because then we all sit around, as we did last December in Birmingham Children's Hospital, and we discuss how the clinics are progressing, the good points, the bad points, how we can improve these sorts of things. And the main objective this year on our agenda has been discussions around developing our database. So this is something that's been long coming, but it's billed as what we would call a patient management system, and it's being built by a company called Certis. And many of you will know that the NHS has not been very successful in pulling together what's called an electronic patient record. And many of you will know that you go to clinic and it's potluck whether your notes turn up on the day, right? So we need to get away from this paper-based system to an electronic system, providing the computers don't go down, of course. However, that's not going to happen because in any time soon, there are at least 10, I'm sure there's more than that, 10 different electronic systems that are running throughout all the countries, all the counties in this country. And just within the 4 clinics that we run within the BBS service, they all have 4 different systems for holding electronic records. And so they don't necessarily speak to each other. So we've decided that what we need to do, at least temporarily for the next few years, is develop our own bespoke electronic customized system, which is this patient management system that we're developing with Certis. And what will it be able to do? Well, what we want to be able to do is to record all of the clinic activity. So every time you come to clinic, we need to know when you're coming, when is the next clinic appointment due. We want to know all of the results you've had when you have your blood test done, when you have your blood pressure done, when you have your weight, height, all those kind of things. We also want to know what medication you're on as well, and we want this all to be in one place, so it doesn't become a mystery to the doctors that you see or the other specialists that you see. And we can also help to implement what we call the care pathway, because we have to now, for rare conditions in this country, there is a government-led remit. It's actually an EU-wide remit that every single patient who comes to these types of clinics has a personal care pathway implemented. So we know exactly what's supposed to happen to you and where you are on that particular path, if you like. Also, this is going to help us not only produce the letters in a more timely fashion, but also keep a record of all of those kind of letters. And eventually we hope to be able to include things like X-rays, ultrasounds, all these kind of images on there as well. And finally, we hope that it will be able to develop what we call a better communications arm. And I'll tell you a little bit more about that in a minute. But the other real opportunity for us here is we're going to be able to develop what we call a dashboard that we can, it's like the dashboard of your car, so you're essentially going to be able to have an overview of the 4 clinics and all the people who are attending. And on a patient-by-patient basis, we'll be able to click on a particular value, if you like, and go in and have a look exactly what was said last time, where you are on the care pathway, what medication you're on, whether your blood pressure has gone up or down, whether your weight has gone sky high, or whether it's just in the same place. You know, all of those kind of things. So it's going to be of a huge value. And it will enable, as I say, other clinicians to be able to look at whichever part of the clinical service you're in, will be able to look at that information. But I want to assure you that it will be highly confidential, that there will only be certain people who are authorized to look at certain elements of the information. But also, what we plan to do is, you know, we're in the 21st century now, and most people have access to the internet, or they have tablets or smartphones or what have you. So Certis is developing this system, so you will be able to access much of this data, again, very safely, through the internet. And so individual patients or their family members will also be able to access their own information, their own appointments, know when the next appointment is, and even communicate with the team. So that, for example, they might want to speak to Kath, our clinical nurse specialist in London, for example, and just talk about latest results or something like that. We will hopefully be able to do that through this system. Okay, we can talk about that implementation later. It's going to, we hope to roll this out for the first testing later this summer, and then get all the bugs out of it so that it should be up and running by the autumn with luck. Okay, so moving on next to another component of our clinic, and that's been the genetic testing service. Because when we put together and proposed the clinics to the Department of Health originally, what was rather unique, they'd never had this for any of the other 69 services in the country, was that we said, look, we really need to do genetic testing because it helps us with diagnosis. And so what I can say is that 419 patients have now been tested from 358 families that attend these clinics. That's by far the largest group of patients in one place on the planet who come to these type of clinics and have been genetically tested as well. And of these, we have a positive genetic test. In other words, the diagnosis has been confirmed through a genetic test in 80% of the families. So contrast this to 10, 15 years ago, when we used to sit here and we used to say, oh, we found one more gene, and we found another gene, and that makes 2 genes. We now have about 19 genes, which are more or less all of them. There's probably maybe 1 or 2 left to be discovered. But we've got to our original goal now, which is finding more or less all of the genes, which means we can now deliver an incredibly robust diagnostic service, which is being used again. People have come to us to ask us to share with them this testing, or even they send samples from all over the world to be tested in the NHS. So we're really pleased about that. What can testing provide for us? Well, we can also test folks within the family who might be carriers. They don't actually have the condition. They might be carrying 1 copy of the faulty gene. So we've done that on a number of people. In fact, almost 100 people now have taken that test. We can also use this test for what we call predictive testing or prenatal testing, and we've done this successfully in about 13 or so cases now. So that means testing the unborn fetus. And so I think this has been a really positive component of the funding that we've had for this particular component of the clinics. Has there been an advantage? Yes, there's a simple answer. So the age range of the people who have been tested in the clinic is from 6 months right the way through to 59 years of age. And we can say that the diagnosis has now been confirmed in 94 patients, almost 100 patients, before the average age, the previous average age of 9 years. So when we published a paper back in 1999 with the help of all of you who were here then, and it was, at that time, it was the biggest study in the world, and it essentially said that the average age of diagnosis was 14 years of age, which is way too late, as you'll know. That came down to about 9 over the recent years, and I think this genetic testing is now reducing that even further, which I think is a positive thing. So we can also say that 24 patients have now been diagnosed before the age of 3, which is the really tricky part. So the very young children, before visual impairment is obvious, has been very difficult to make that diagnosis. So this has been a major advance in this particular area, particularly for the younger child. I won't go into that. Phil earlier mentioned that the society was represented at the Cilia Conference in 2014 in Paris, which was a conglomeration of about 400 attendees from all over the world. It's a scientific and medical conference relating to ciliopathies, of which Bardet-Biedl syndrome is one of those, a big umbrella group of diseases. And this just shows, because we started with the help of the society back in 2012, the very first of this series of international conferences back in London at our institution. And since then it has grown, and it's now a biannual event. So the next one will be in 2016 in Amsterdam, to which you're all invited. But it just shows the continuing interest in this whole area. And what's quite clear is that there's a shift now towards looking at treatments in this area, which is something we couldn't even talk about 3 or 4 years ago. So that brings me really on to the activities that we've been undertaking in my research lab down in London at UCL. So for those of you who don't know, I'm based at UCL and at Great Ormond Street Hospital, and also at Guy's Hospital as well for some of the time. And we've assembled over the years a very active lab, which we call the Cilia Disorders Lab. We have about 14 people actively working in various areas. Each of these people have to be funded through grants. We don't get given money from anywhere whatsoever. We have to apply competitively for grants, and that's been really tough over the last years because all of the recession and so forth only really is starting to hit us now, this many years later, in the funding community. But nevertheless, we've been able to keep the lab going, and the majority of people within that lab are working actively on not only understanding what the causes of Bardet-Biedl syndrome are, why each of the problems develop, but also now a significant group of them are working toward finding therapies for this condition, finding treatments. So I will spend a couple of minutes talking about that. So we have been in the past interested predominantly in, well, finding genes was our original target, but that's been so successful with other groups around the world that we don't have to do that anymore. And as I say, a lot of people are still working on understanding how the disease appears and what makes it go in one direction or appear in different members of the family but not other members of the family in a particular way. But what we're really trying to focus on now is thinking about what potential therapies we could be developing. But as I mentioned last year at this conference, we do have some elements on our side. In particular, the pharma industry, the pharmaceutical industries, are really, really interested now in rare diseases. They couldn't care less about 5 years ago, actually 3 years ago. They were more interested in the diseases that more commonly affect the majority of people in this country, you know, high blood pressure, heart disease, asthma, these kind of things. Not saying that they're not important conditions, but that's where they saw their revenue stream coming from. That's where they saw the money being made, okay? But there's been a bit of a crisis in their world, and that's that the new blockbuster drugs aren't coming anymore. And so what they're doing is they're turning their attention to conditions like Bardet-Biedl syndrome, because they see these, because, you know, hypertension's part of the problem, high cholesterols, you know, all of those kind of things that are commonly seen in the general population. And what the pharmaceutical industry now realizes, which we've been telling them for many, many years, is that if you get involved in our condition, then you might be able to understand better other more common conditions, but also provide those new drugs for those conditions. And that's where they're really engaging with us now. So that's all positive. What types of therapies are we talking about? So we can talk about, and I did last year actually talk about developing medicines, that's drugs, potentially for Bardet-Biedl syndrome. I'm not going to talk about that this year, but last year we talked about, you know, using things like zebrafish, for example, to screen drug libraries and so forth. And what we're more focusing on, perhaps, or want to talk about a bit more this year, is either gene engineering or gene therapy. And we are actively engaging in this area at the moment, particularly in gene therapy. There will be a talk, obviously Jim's going to give us a much more detailed talk about this, but just to touch on this area. So gene therapy, the idea of gene therapy is not to replace the faulty gene, but to augment it. So to insert, if you like, a normal copy of the faulty gene. And by doing so, you hope that that gene, the normal copy of that gene, will produce the protein, because that's mainly what genes do, will produce the protein that is missing or is faulty. That's what you want to do in each cell in the body, or the cells that matter. So that means there are, you know, how do you deliver this good copy of the gene? So the first thing to decide is how are you going to do this, or what are you going to target? And so the obvious one, and we've talked about this before at this conference a number of times, is the obvious one is the visual loss. Is it possible that we could prevent or slow down or delay the visual loss in Bardet-Biedl syndrome by inserting copies of a good gene, because there's clearly faulty genes there, or a faulty gene. And so in order to do this, what we've started to do, and I've told you many times before that in our lab, whether you agree with it or not, what is incredibly valuable to us is these so-called animal models of disease, either zebrafish, or what we work on a lot, or mice. So we've made a number of mouse, what we call mouse models of disease. So the little mouse has Bardet-Biedl syndrome. And we've been very lucky because those mice emulate, they recapitulate the human form of the disease very, very well. They develop the increase in weight, and they become visually impaired. Okay, that gives us therefore a really good starting model on which not only to test why this has happened in the first place, but also to test either new drugs or, in this case, the potential for gene therapy. And so Victor Hernandez is in the audience here. Where's Victor? Stick your hand up. So he's been working hard in the last year or so to define, to have a look at our mice that have Bardet-Biedl syndrome. And in this case, what I'm showing here, I'm showing a picture. Don't get too excited by it because it's all a bit too detailed, and you can't see the detail here. But to see what is actually gone wrong in the mouse eye. And what we're showing here is the retina, the layer at the back of the eye that captures the light and sends it obviously to the back of the brain, the image. And those of you who can see the screen will be able to just see these sort of differences in height. And that's all I want to point out, is that essentially we're showing here a normal mouse eye compared to an eye from a mouse that has Bardet-Biedl syndrome. And the one that has Bardet-Biedl syndrome has a much thinner retina, has a thinner layer at the back of the eye. And that's because the key cells, the so-called photoreceptor cells, have started to die so that the layer has got much thinner. So that's helpful for us because that can be the starting point for not only trying to understand why that's happened, but if a therapy of some kind is going to work, then we're going to want to be able to make sure that this is retained, that the height or the thickness of the retina is maintained and that it's functioning normally. So that's the intention. So how would we do this, or how are we doing this? So with colleagues at the Institute of Ophthalmology, Sandra Smith and Robin Ali's group, Victor's been preparing the normal gene. Actually, we're using a human gene in this case, but inserting it into a mouse. And it gets injected into the back of the eye. And I think Jim will tell us a lot more detail about how that's done. But what Victor does is he injects just one eye of the mouse and keeps the other eye untreated. So in a mouse that's got Bardet-Biedl syndrome, you've got what we call an internal control, something to compare the treated eye with and then the untreated eye, if you get me. And so it's very early days, but we have some promising results, and I'm sure Victor will talk to you later on if you want to. For example, when measuring the ERG, most of you will have had an ERG. Remember when they used to put those little electrodes on your face and on your forehead and so forth? Well, you can do that on the mice as well. And it's looking promising in that the early results suggest that in the treated eye, it seems that the activity in that eye is retained. But as I say, we've still got a way to go. And I'm not going to steal Jim Bainbridge's thunder because he's going to talk a lot more about the advances they've made over the years and how they're the real experts in the world in this particular area. One other area with regards to gene therapy that we're actively seeking, which is quite unique to my mind, and that is around the fact that Bardet-Biedl syndrome doesn't just affect the eye, does it? You all know that. It affects many other parts of the body. In fact, we don't know an organ in the body that Bardet-Biedl syndrome doesn't affect. And so therefore, it truly is what we call a multi-organ disease. Therefore, if we're going to do gene therapy, wouldn't it be better, if possible, to be able to give that therapy through a vein, for example, an injection, and then it reaches all the other parts of the body that other genes don't reach. So that's what we are aiming toward. And so with some colleagues again at UCL, Dr. Ahad Rahim and also Professor Simon Waddington, they've already developed the proof of concept for this study going forward. So instead of focusing on just one organ, as I've told you about within the eye, what they've been able to do is show that by attaching a colored tag to the virus that we use, and you'll hear more about the virus later on. But the virus is used to carry the gene into the cells, okay? But what they've been able to do is inject virus into mice, and in doing so, show that that virus is getting to all the parts of the body that we need to get to. So I'm showing a picture here, which is doesn't show up too well. At the top here, there's a mouse that has been injected with this virus, which has what's called a green fluorescent protein tag on it. So when we shine a UV light on it, it shows up where that, where the virus has gone, but we would hope also where the gene has gone. And it lights up all over the body, essentially. And then the pictures at the bottom here are showing different organs. So for example, the kidney lights up like a Christmas tree, which means that the gene is getting to those parts of the kidney that we would like to see it get to. And we also see it getting to the eye as well. So the beauty is that with this particular system and using these particular viruses, they're crossing the blood-brain barrier. So lots of big molecules can't normally cross the blood-brain barrier, but in this case, they can do. So we want to exploit that opportunity to see whether we can deliver BBS gene to all the different parts of the body. And we can talk about this later on. Okay, the very last part I want to really just talk to you about is an area which is very new. Very little is published on this area. But I can say again, with the help of all of you over the years, that the success story in the research that has been published around the world from multiple labs that work on Bardet-Biedl syndrome has been focused on understanding why are there kidney problems, and why is there visual deterioration associated with Bardet-Biedl syndrome. But we all know that some folks do have some learning disabilities, others have some problems with memory, for example. And so those aspects of the brain have not been studied before in Bardet-Biedl syndrome. But, and this is an area we really want to advance. So in our lab, Sonia, and I showed you one of these, or touched on this last year, but Sonia, who's also here in the audience, put your hand up, Sonia, down here, has been working really hard on understanding what's going on in the brain. And she's predominantly been using the mouse again, because that's a major value to us. And what she's been able to do is work out at least the early start of what might be going on, is what's going on in the brain cells in the brain of these mice that have Bardet-Biedl syndrome. And what's quite clear is it's to do with the connections between the brain cells. So some of us, not all of us in this room have brain cells. But those of us who do, and I don't count myself as one of those, but those of us who do have them will have, on each of our brain cells, we'll have all of these little finger-like appendages sticking out, which are called dendrites. And on those dendrites, there are little buttons on them. This is all microscopic. You wouldn't be able to see these without the aid of a microscope. But these buttons are really important. They form the connections between the next brain cell, and then that one forms the connection with the next brain cell, and so forth. And what's really important, it's important not only the type of buttons that you have, or synapses are what they're called, but the number of those synapses as well as the function of those synapses. So in short, what Sonia's been able to do is to determine that in the Bardet-Biedl syndrome mice that we've looked at, not all of them, but several of them, have reduced numbers of these buttons. So in other words, there's less synaptic activity, less connections, or less opportunity for communication between those cells to happen. So this is the very first time this has ever been discovered. It's not actually been published yet, so you're all sworn to secrecy in this room. But the point being is that this gives us a starting point to be able to think about how we might even be able to modify some of these learning difficulties. And so Sonia's actually gone one step further and thought about how you might actually do this in the simplest form. And this is a really active area in neuroscience at the moment, particularly in dementia, which is one of the big unsolved problems, but a major economical problem in the whole world, and has now become a focus for research attention. But it's been known for a number of years that simple activities such as exercise, increasing exercise on a daily basis, can stave off dementia and can improve our learning ability. So what she did was put our little mice onto a treadmill. Well, it's actually a wheel, exercise wheel, and they love going on the exercise wheel. And what she's been able to show is that those mice that actually exercise, compared to the ones that are deprived of exercise, so we give them a television to watch, and they got a nice couch and all the rest of it, and they've got Coke and chips and what have you, and then the other ones, go on the treadmill, and it's the ones that are on the treadmill that she's been able to show that the number of buttons, the number of synapses has increased back to normal in those mice. So what we're hoping is that this is something that we can also benefit from as humans, all of us, but also the Bardet-Biedl syndrome community might also be able to do that. So what we're hoping is to get all of you out there, or at least some of you out there, and onto an exercise program. And I'm going to hand over to Elizabeth now, who's going to tell you about a workshop this afternoon where you can take part in some of these studies that are going to help us design the program to go forward to see whether this is going to make a difference at all to all of you. So I'll hand over now, and then I think we can go move on to the next talk. Have you got a mic on you? Could we have that one on?
You can use this one.
Okay, even closer. Better? So I'm one of the clinical genetics doctors in London, and I think I've met quite a few of you in the clinics over the years now. And I just wanted to briefly talk to you about this workshop we're doing this afternoon. And it's a workshop looking at learning and memory in Bardet-Biedl syndrome. And we wanted to look into this in particular, really because of the feedback we've had from you over the years in the clinics. We know from speaking to many of you and to speaking to parents and carers that many of you have excellent long-term memories, but quite a few people have difficulties with particularly short-term memory and often attention as well. And that has a knock-on effect on the ability to learn new skills and also learning at school. We know that most people who have BBS, who have learning difficulties, usually have mild learning difficulties. And that means that actually most people have the potential to work and live independently. And so this has become an important issue that's been brought up many times over the years. So I just wanted to tell you very briefly about what we know about this area already. And actually the answer to that is very little. There was a big study a couple of years ago in the States where they tried to get a little bit more of an understanding of this. And what they found was pretty basic. It was what you guys have been telling us for the last couple of years, which is that really being able to maintain attention for longer periods of time seems to be one of the key issues. There have also been a couple of other studies where people have looked at different areas of the brain, and what they found is that people who have BBS often have slight differences in their brain structures. But how that relates to your ability to remember things well and ability to learn new things, we haven't really clarified yet. So what is it we're trying to do this afternoon? What we'd like to do really is, first of all, to get a really good sense of the spectrum of differences in terms of ability to maintain attention in particular, and also learning new things and sort of impulsiveness. And the idea here is, if we understand this better, hopefully we can progress to developing some treatments or improving this in the future. So how are we going to do it? Well, it's through this workshop this afternoon. The way we do it is it's through a fun computer game. It is not scary and it is not difficult. And we know this because we tested the computer game last night on a couple of members of the BBS clinics who had all had a lot of pints. So please come along to the workshop and beat them, okay? So because of licensing agreements, we can only do one test at a time. So what we're doing in the meanwhile, while each person goes in to do the test, is that I will be running a question and answer session. And really, I'm going to kick off by talking about some of the questions that come up quite frequently in the clinics, and also what I hear comes up on the BBS Facebook group. Things like, what does it mean to have BBS 1? What does it mean to have BBS 10? I have BBS, what does that mean for my children? That sort of thing. And then I will leave it up to you really to answer any questions you want to. And that's it for me. I'd better hand back to Phil. Thank you.
Okay, thanks, Elizabeth. Just one final word I want to say, or message rather, is from Helen May Samira, who many of you will know has been a staunch supporter of this society over the years. For those of you who do know her or can't remember because of the short-term memory issue, she originally did her PhD in my lab, but moved to the States about seven years ago, I think, and has really forged her career still working on Bardet-Biedl syndrome. And just 3-4 months ago, moved to Germany. She speaks fluent German, married an American, so took him along too, and has started her own lab there, which is a major advance for her. But she wanted to apologize for not being here today because she's in a state of pregnancy. In fact, she's five days overdue, so I said, well, it's conference day today, so you're likely to give birth today. So she wants to say hello, and we send her our best wishes. So that's it from me, and we will move on swiftly now to our next presentation. Elizabeth, Elizabeth's agreed, I don't know whether willingly, but has agreed to co-chair the day with today, so you don't get too bored of me.
This audio recording was taken live at the LMBB Society Conference in Northamptonshire, 18th of April